everybody, I mean, I see people nodding and I see people's faces, and that's great. And I can also look outside my window and it looks like it's a beautiful day outside. So what's not to like about a Wednesday? Let's see, let's let one more person in. The scientist that I'm going to highlight today is Dr. Ariane Baldomero, and she is a pulmonologist. So we're hopefully going to get into talking about the respiratory system today. So I wanted to pull up a lung specialist. So her specialty is pulmonology, and then she also has subspecialties in general pulmonary medicine, critical care medicine, pleural disease. When I saw her picture, I thought, how can this person possibly be a doctor? She looks so young. But yeah, she is. And so you guys can be doctors too. Or you can get into some other kind of science. All right. So that's our science person of the day. So let's move into talking about giraffes. I promised you that we would talk about giraffes. And so I want to show you a video to begin with. So let's look at this video. And actually, I'll change my sound settings so that you can hear it. So it's kind of a big effort for them to get down and drink. And what I want you to watch is how fast they come up from drinking. Whoa. Can you imagine the head rush? Isn't that impressive? All right, enough of that. Let's see, let's find our PowerPoint again. So the question I have is, why when they bend their necks down to drink? So I would like you to send a private message to your lab partner to answer this question. What would be some structure, maybe function? What would be some anatomical, physiological adaptations that giraffes have so that they don't faint when it's not really when they're bending their necks down, but when they come back up after having a drink. So send a private message to your lab partner, and then I will call on somebody. I can think of actually several reasons that I will highlight with you. So think about what happens for us that keeps us from fainting, and then kind of think about what it must be for giraffes. Okay, so my first person today is, let's see. I'm going to call on Kylie. What do you think? What are some anatomical, physiological reasons why giraffes don't faint when they come up from a drink?
Does it have anything to do with like where their arteries are located?
There are some structural features about their arteries, but their arteries are in the neck just like ours are. Um, do you have any other ideas?
Um, I mean, I think it has something to do with how fast they bring their neck up, but that doesn't. That's not really what you ask. So.
Well, so they do come up really fast, and I think that's really important for them. They have to be able to raise their head really fast so that they can run away and so they can escape from predators, right? So, what are some anatomical and physiological features that enable them to do that without fainting? So, think about their heads way below their heart. So, you can imagine that their head is going to get a lot of blood, and then they raise their head really suddenly, and then suddenly their head is way above their heart, like 10 feet above their heart. And so suddenly they just don't have as much blood in their head. Let me call on somebody else. I'll give will be equal opportunity today. Um, how about Kayla? Do you have anything that you'd like to add?
I'm not really sure.
Okay. Let me just throw it open to anybody. What do you think enables giraffes to not faint, especially when they're coming up after a drink? Who has an idea?
Um, I thought of a few things. Perhaps the giraffe's heart is just specialized to pump crazy amounts of pressure, so it can just immediately send blood right back up the neck when it starts falling down. Or maybe I thought there's like a valve that keeps the blood from falling super hard towards the head, and then maybe a valve that keeps the blood from just leaving the brain when the head comes back up. Like keep it suspended there. Mm.
Okay, you got my first two. So the heart of giraffes does generate tremendous systolic pressure. So what would be the systolic pressure in a human? Anybody? When you get your blood pressure, what's the upper number?
130, 170.
Yeah, somewhere around 120, 130, 130, 135 is getting a little bit high. So we kind of want to have it about 120 over 70-ish. And so the giraffes generate twice that much pressure, which is a crazy amount of pressure. And that is just because their heart is bigger and really strong muscle, and it can generate just a ton of pressure. And then most mammals do not have valves in our jugular veins because the blood in the jugular veins is flowing downward toward the heart. But think about when the giraffe bends down, the giraffe doesn't want all that blood to go rushing towards the head. And so it's got valves in the jugular veins that prevent the blood from flowing toward the head. So that prevents it from having too strong of pressure when it's got its head down. So that's kind of an unusual adaptation. And then also at the base of the brain, all mammals have kind of a network of blood vessels at the base of the brain, but it's even more effective in giraffes, so that they have a way of sort of shunting the blood so they don't get too much blood pressure that goes into the brain, especially when their head is down. And then Another thing that really helps with them maintaining high enough blood pressure to get the blood up to the head when their head is up high is that they have super thick and tight skin in the legs. It's like they're wearing pressure stockings all the time, and that prevents the blood from just pooling in the brain. I mean, in the legs. So these are a couple of kind of specialized adaptations that giraffes have. I really think animals are cool. I really think that just the different adaptations that different animals have are really cool. And so I like to talk about more than just humans, because I think, God was really creative with the way he made all different kinds of animals. So I like taking advantage of opportunities to share that with you guys. All right, let's talk about blood flow in the capillaries. Before we move on to the respiratory system. And so you know that the capillaries are where all the exchange happens between the blood and the tissues. And so it's really important to regulate blood flow in the capillaries, how much blood is flowing through the capillaries. We've already talked about this a little bit in terms of how we regulate blood pressure. So blood pressure is primarily regulated by vasoconstricting or vasodilating the arterioles. And that regulates the amount of blood that's going through capillaries in different parts of our body. So you can vasoconstrict the blood vessels, the arterioles that are leading to the skin, and that keeps more of the blood in the core of your body, for example. Or after you've eaten a meal, you can vasodilate the arterioles that are taking blood to your digestive organs, and then that helps you send lots of blood to take nutrients from the digestive organs to other parts of your body. But blood flow in the capillaries is also regulated by little tiny sphincters, little tiny circular muscles at the beginning of the capillary beds. So in this, the upper figure here, part A, shows these little sphincters. And in part A, the sphincters are relaxed. So the blood is allowed to flow through all of these capillaries. And so these capillaries would be actively exchanging materials with the cells around them. But if you want to not send as much blood through the capillaries, your body can close off these sphincters, and then the blood just bypasses the capillaries and goes through these thoroughfare channels. You can see this labeled up here. And the blood just goes right through the thoroughfare without going through most of the capillaries. So it reduces the blood flow through the capillaries, probably doesn't completely shut it off. But this way you can, this is a way of regulating blood pressure and also just regulating what parts of your body get more blood so that exchange can happen. Now, at the beginning of the capillary, so blood is coming through the arteriole that's coming from the arteries that came from the heart. And at the beginning of the capillary bed, so this kind of network of capillaries we call a capillary bed, at the beginning, the blood is going to be under pretty high pressure, right? And so we have high blood pressure at the beginning of a capillary at the arterial end of the capillary. And that blood pressure diminishes as the blood moves through the capillaries. So at the venous end or the venual end of that capillary, there's much lower blood pressure. But notice that there is an opposing force that's shown with the blue arrows, and that is the osmotic pressure. And this osmotic pressure is based on dissolved substances in the blood. So you can see that the blood cells, the red blood cells are shown here. They do not leave the blood vessels in most cases. Sometimes a white blood vessel, white blood cell can squeeze its way out of a blood cell. This happens if there's an inflammation and the white blood cells are needed to fight some sort of invading material out here, but mostly the blood cells stay inside the blood vessel. And as you read about, there are also lots of proteins in the blood that are too large to diffuse out of the blood vessel. And so because of these proteins and because of the blood cells, there is a gradient, an osmotic gradient that would draw water by osmosis from the interstitial tissue into the bloodstream. So water tends to move from an area where there's a low concentration of materials to where there's a high concentration of materials. You can think of it as kind of evening out the concentration level. So if there's a high concentration of solutes in the blood vessels, then water would tend to move by osmosis into the blood vessel. And that is what these blue arrows are showing. So overall, you can see that there will be a net loss of fluid, of water, based on the high blood pressure, and the osmotic pressure draws some of that water back into the blood, but not all of it. So there's going to be an excess of fluid loss from the capillaries. So not all of the fluid that leaks out is going to be able to be drawn back in by osmosis. So the question is, what happens to that fluid? If that fluid were not eventually returned to the bloodstream, you would end up with your whole body being swollen because you'd have all this fluid that had leaked out into your tissues. And in fact, if that fluid doesn't come back into the blood eventually, then you get what's called edema, or swelling. So what's the normal process for returning this fluid to the bloodstream? Give your answer to your friend in a private chat. Your friend, i.e. lab partner. What happens to this fluid that's lost from the capillaries? How does it get collected and returned to the bloodstream? All right, let's see. I'm going to call on Nate Ingstrom for this one. What do you think, Nate?
Does it like get recirculated by this like the circulatory system or something?
Well,
I don't know.
it has left the circulatory system. So it's out there in the interstitial fluid, and not all of it gets absorbed back into the blood, but there's another type of circulatory system that is, so you're close. And the answer really is that it gets collected by the lymphatic system. So remember when we talked about the villi in the small intestine and how there are both blood capillaries and lymphatic capillaries, or lacteals? There are actually lymph capillaries, which are shown in green in this diagram, all over your body. And so they are able to recover some of the leaked fluid and also leaked proteins. And these lymph vessels have valves in them, so they collect the fluid, and the fluid, as your muscles move, it squeezes on them and it forces the fluid back up toward the large blood vessels in the neck area. And so large veins at the base of the neck is where this fluid is going to dump back in. So this is showing kind of some body cells, and it shows a capillary bed. So the arrows are showing the flow of blood through the capillaries. And then the black arrows show fluid that leaks out of the capillaries and goes into the lymph vessels. And then here's a figure from chapter 43 that shows a bigger view of the lymphatic system. So you've got lymphatic vessels all over your body, and they're collecting fluid and some proteins, and they filter that fluid through the lymph nodes. So over here is a diagram of a lymph node showing how lymph comes through, and there's lots of white blood cells in these lymph nodes. And you learned in your reading that the white blood cells are involved in body defenses. So this kind of cleans the lymph as it passes through and continues on to these large lymph ducts. And these large ducts dump into the large veins at the base of the neck. So the jugular vein, or the not the jugular vein, the brachiocephalic veins. But you don't really need to know that. Okay, so that's the role of the lymphatic system. All right, so let's do a multiple choice question, which you can answer by a private chat to me. A, B, C, or D. Fluid is filtered from blood as it enters a capillary due to blank, but fluid is reabsorbed as blood exits a capillary, reabsorbed back into the capillary due to blank. Okay, I've gotten a lot of answers. I'll give you 5 more seconds to put in A, B, C, or D. At this point, if you don't know, you can just guess, and that'll be good enough to give you your participation credit. And so some of you didn't know, but the correct answer is C. It's the blood pressure from the heart that forces fluid out, and then it's osmotic pressure due to the high concentration of proteins in the blood plasma. Let's maybe go back to that picture that showed this. So you can see high blood pressure at the arterial end of the capillary, and the osmotic pressure is the blue arrows, and that is due to the proteins that are in the blood. Okay. So I'm not going to cover the constituents of the blood in lecture. You read about that for your assignment today. You learned about the red blood cells, the white blood cells. You learned about what the plasma carries. You learned about a little bit about the formation of blood cells. All the blood cells are formed from stem cells in the bone marrow. And one person asked an interesting question about what a blood cancer would be. And if you have a cancer like leukemia or some other blood cancer, that is really a cancer that happens in the stem cells in your bone marrow. So a leukemia is the stem cells that are leading to certain white blood cells. They are dividing more than they should, and that is increasing the amount of white blood cells in your blood. But I do want to go over this skills exercise. So I'm not going to cover most of what's in the section about the blood components, but I do want to talk about this skills exercise a little bit because some of you guys had a hard time interpreting these graphs. And so you were asked to calculate the percentage of people that had in the study group and in the control group that had a plasma LDL cholesterol level less than 100. So the way you do that is you would just count these bars that are to the left of the 100 line. So this is plasma LDL cholesterol on the x-axis, and on the y-axis is the percent of individuals. So 30% of individuals in the study group had a plasma LDL between 50 and 75. Another about 23% had an LDL between 75 and 100. So if we add that together, we get a little bit more, that we get about 53% plus another 4% maybe is about 57% of individuals less than 100. So more than half of the people in the study group had an LDL level less than 100. Whereas in the control group, and these are people where the PCSK9 gene was working, the majority of those people had an LDL level greater than 100. As you can see, most of these people were higher than 100. Only about 13, 14, 15, 16, maybe about 17% of people had an LDL less than 100. So does that sort of help you guys see how to interpret that? I see like 2 people nodding, so that's good. Okay. So that's the main things that I'm going to cover. I'm glad that you were able to read about cardiovascular diseases, but we want to stay a little bit more focused on energy and metabolism. So, so we're talking about the circulatory system because it's carrying the nutrients that came from the digestive system to the body cells. And now we want to shift our focus to thinking about the respiratory system and gas exchange, because your cells not only need nutrients that are coming from the digestive system, but your cells also need oxygen that's coming from the respiratory system. So we're going to shift our focus to talking about this. You have an assignment that's due on Friday so that you can read about this and kind of reinforce what we covered today. So our learning objectives for the first part of the respiratory system is have to do with respiratory surfaces and respiratory organs. And so in thinking about this, we also want to talk about, think about air and water as respiratory media. And then we'll look at structure and function of respiratory surfaces. And this is going to depend on whether animals are trying to get their oxygen from the air or whether they're trying to get oxygen from the water. Kind of depends on where they live. And then we'll look at the process of ventilation. So work with your lab partner to define these terms. Take turns. If one of you sends a chat defining respiratory medium, the other one can respond with the definition of a respiratory surface. So ready, go. All right. So I'm going to call on, let's see, Kayla, I'm going to give you another chance. Kayla, would you like to define respiratory medium or respiratory surface? Kayla, there she is.
Respiratory medium is the source of oxygen.
Yes, respiratory medium is the source of oxygen. So it's either going to be air or water. And then the respiratory surface is the part of an animal's body where the gas exchange occurs between the respiratory medium and either directly with the body cells or with the circulatory system, which is going to carry that oxygen to the cells. So the medium is the source of the oxygen, which is either going to be air or water. And then the respiratory surface is where the gas exchange occurs. So let's focus in on the two main kinds of respiratory media. And the first is air. This is what we're used to because we breathe air. So the advantages of air as a respiratory medium are that the oxygen is quite plentiful in the air. Oxygen makes up 21% of the atmosphere. And also air flows easily. We would say that air is very non-viscous. It's thin. It flows easily through the air. And also through the respiratory organs of animals, lungs, trachea, book lungs in spiders, etc. Water, in contrast, always has less oxygen than air does. So oxygen can dissolve in the water. So there's oxygen molecules like mixed in with the water molecules, but the oxygen concentration is always going to be lower in the water than it is in the air. And if the water is warm or salty, it's going to hold even less dissolved oxygen than cold fresh water does. And then the second disadvantage of the water is that it is much denser than air. We don't think of water as being viscous. So when you think of the word viscous, you usually think of something like molasses, that if you pour it, it flows really slowly, or honey. So water is not very viscous compared to molasses, but compared to air, water is much more viscous. And there's a table in your reading assignment for Friday that shows the viscosity of air and water. And water is way more viscous than water, many times more viscous. So you can see why it would be advantageous if we look back in the history of the earth. If animals used to live only in the water, you can see how it would have been an evolutionary advantage for some of them to start living on land, because it would have been easier to get oxygen. But there are some disadvantages, and we'll talk about that when we talk about respiratory surfaces. So the thing to understand about respiratory surfaces is that this is where gas exchange happens. And so in order for gases to diffuse across a membrane, there's always a membrane. And in order for a gas to diffuse across that membrane has to stay moist. So this is one disadvantage of trying to breathe air, is that air is pretty dry and it dries out your respiratory surface. So gases move entirely by diffusion. Your cells do not pump gases using active transport. They move entirely by diffusion. And diffusion is proportional to the surface area available. The more surface area available, the more molecules can cross through that surface area. And gas diffusion is inversely proportional to the distance that that gas has to move. So if you've got a surface and a gas molecule is moving from above this surface to below this surface, the thickness of this surface is really important. Gases are gonna, like the thicker that membrane, the farther the distance the diffusion has to be, and so it's really going to slow down. So as you can imagine, then a respiratory surface is always going to be really thin, and it's going to have a high surface area, and it will have to be moist. So let's talk about the different kinds of surface areas, the different kinds of respiratory surfaces that different animals have. So first, some animals that have very simple bodies, like the sponges, they are able to just get oxygen through their epithelium. So sponges and cnidarians, like the hydras, they have very thin body walls and a very thin epithelium, and so gas can just diffuse across that epithelium, and all of their cells are very close to that epithelium or close to the air or water that's surrounding them, and so they can just rely on diffusion. Amphibians, many amphibians also rely at least partially on their skin for respiration, and earthworms also do. So earthworms and amphibians, they usually have to keep their body surface moist so that they can do gas exchange through their skin. So amphibians are large enough, have large enough bodies that they also have lungs, and so they have to rely partially on their lungs and partially on their skin. Earthworms don't have lungs at all, so they rely only on their skin. Most organisms need a specialized respiratory organ. And why is that? Answer this question in a private chat message to your lab partner. Why do most organisms need a specialized respiratory organ in addition to their skin? Okay, Ben Terpstra, why do you think most organisms need a specialized respiratory organ? Actually, I'm going to check my participants. Looks like Ben's not with us today. So let's go with, next on my list is Lily.
Is it because of like the volume of like their body, so they need it to pump more oxygen to
Yep,
different
that's
places?
exactly right. So it has to do with surface area to volume, right? So the skin does not have enough surface area to supply the entire volume of the animal, like Lily said. So that's the big thing. It's surface area to volume. And so the solution that animals have, since the skin doesn't do the job, is that they're going to have a respiratory organ that has either extensive folding or branching so that there's a lot of surface area for gas exchange. And in some animals, this is an external organ, but for land animals, usually this is internal so that it doesn't dry out. So let's see. So if you are if you're an aquatic animal, you can use gills. And so these are outfoldings of the body surface that are suspended in the water. Sometimes they're covered up by a carapace, like with this crayfish. There is a shell that covers over the gills to protect them from damage, but the water can just kind of go underneath the gill cover. And the cool thing too about crayfish is that their gills are actually attached to their legs, which I have a picture here of a crayfish that's partially dissected, and you can see that the shell, the carapace that covers the gills are shown in number 2 here, and the carapace has been peeled away, and so you can see that the gills are attached to all of their walking legs. So number 1 shows the base of all their walking legs. So every time they move their legs, they're helping to ventilate, they're helping to bring fresh water into their gills. So how does an aquatic animal maintain partial pressure gradients across its gills? Okay, so you have not read about partial pressure gradients unless you have read ahead, which I'm sure a few of you had, but many of you haven't. So you will, for your assignment on Friday, you'll read about partial pressures. And you can think of partial pressure gradients as similar to like concentration gradients. So the partial pressure of a gas is the pressure that is exerted based on only one gas in a mixture of gases. So this question is kind of like, how does an aquatic animal maintain a difference in partial pressure between the water and its blood? Remember that gases only move by diffusion. So that means they're going to move from an area of high partial pressure to an area of low partial pressure. So in order for gas to diffuse from the water into the blood, there has to be a partial pressure gradient, a difference in partial pressure between the respiratory medium and the blood. So an aquatic animal has to be constantly ventilating its gills. It has to be constantly putting fresh water past its gills. And so I'm going to show you, I think I'll see if this link to this giant octopus works. I'm not positive it does. If it does, we can see a giant octopus and how it ventilates its gills.
When school isn't a place you have to be, why not go to school where remote learning can be as remote as you want? Here are some plants I found hiking.
Let's skip the ad.
Hi everyone, I am Candace Reed-Rose. I'm an aquarist here at Monterey Bay Aquarium. If you're here, you're probably wondering, how does an octopus breathe? Octopus and most cephalopods kind of have this two-part body, the mantle being the head region, That's where all of their organs are going to be stored. The siphon, you can see it lives right by their mantle. When they bring water into their bodies, they're going to bring water into their mantle where they have their gills. They're going to flush water over their gills so they can breathe and get the oxygen from the water. And then the water is going to come out through the siphon, and that's also how they expel their waste. That's how an octopus breathes. Thanks for watching this video about giant Pacific octopuses. Be sure to subscribe to our page for more information on giant Pacific octopus.
So I think that's kind of cool. So the giant octopus, as you saw, has a siphon. And I'm going to plug my headphone back in here. So the in-current siphon that draws water into the inside of its mantle cavity, and that's where the gills are, as she explained, and then water goes out through the excurrent siphon. And so the octopus is constantly doing that, bringing water in, sending water out, bringing water in, sending water out. Clams
I have a question,
have a,
just out of
yeah, go ahead,
curiosity.
Chloe. Say that again, Chloe.
Oh, I was just saying that I have a question. So I know that certain types of octopi can come out of water for periods of time. Is that just like the holding their breath, or do they also have gills that can work with air?
Oh, that's a really good question. So maybe what they do is they kind of hold some, no, I wouldn't think that they would hold water so much in inside their mantle cavity, but maybe what happens is their mantle cavity stays moist enough that they're able to do gas exchange with the air over the gills. As long as those gills don't completely dry out, they could still do some gas exchange. So I'm just kind of guessing. I don't really know, but that's a really that's a really interesting question. Okay, so I'll post a fingernail clam video later. And then let's look at, well, you know how fish ventilate their gills, because you guys have been studying that up close and personal in the labs. So you know that fish are going to open their mouth and then water goes through their mouth and then out their gills. So I don't have to show you that video. But I'll show you a couple of questions. I'll show you a couple of figures here. So this shows the pathway, and then we'll zoom in on this picture and we will look at the structure of the gills and how there is counter current exchange between the water moving through the gill and the blood moving through the gill. So you can see these blood vessels. Now think back to the single circulation of a fish. Is the blood coming from the heart of a fish going to be low in oxygen or high in oxygen? You can answer that question to me in the chat. Is the blood coming from the heart going to be high in oxygen or low in oxygen? Okay, 5 more seconds to either say high or low. Is the blood coming from the heart going to be high in oxygen or low in oxygen in a fish? All right, so let's look at the single circulation of the fish. Remember that the blood in the heart has come back from the body, and so it's very low in oxygen, and that blood is going to go into the gills. So blood coming from the heart going through an artery is going to be very low in oxygen. So the blood going into the gills from the heart is going to be low in oxygen. And so that is shown in the blue vessels. So oxygen poor blood, this is coming from the heart, is going to go into one of these gill arches. And you can see from the way this is structured, so we're going to zoom in on just a tiny part of this, but the blood is going to go in on the side away from where the fresh water is coming. So the blood comes in to the gill arch, and then it goes through a through a lamella, which is a layer in the gill filament, and the blood moves kind of opposite the direction that the water is flowing through the gill. So this means that there can be a counter current exchange. Counter current is when you have two fluids that are moving in opposite directions, water going this way, blood going this way. And the reason that this works and the reason that this maximizes gas exchange is because all along this membrane connecting the water to the blood, all along this membrane, there is going to be a higher partial pressure of oxygen in the water than there is in the blood. So the water coming into the gills has a relatively high partial pressure of oxygen. And as some of that oxygen diffuses into the blood, the partial pressure of oxygen in the water decreases. And then in contrast, blood coming into the gill has a low partial pressure of oxygen. And as it receives oxygen that is diffusing from the water, the partial pressure of oxygen increases. So all along this exchange surface, there is a higher partial pressure of oxygen in the water than there is in the blood. And so there's always diffusion all along this exchange surface. So you can see this maximizes the gas exchange efficiency. And we saw a similar countercurrent exchange, but of heat remember, in the goose foot. So you had warm blood coming from the heart, and it was exchanging heat with the cold blood coming back from the foot. And all along that countercurrent exchange surface, there was going to be movement of heat from higher heat to lower heat. So this is a similar principle, only this time what's being exchanged is oxygen. And at the same time, carbon dioxide is going in the opposite direction. The blood that comes into the gills is going to be low in oxygen, but it's going to be high in carbon dioxide. And so it's going to exchange carbon dioxide into the water all the way along there. If you're having a hard time getting your mind around this, I recommend that you watch the animations in your textbook. I think they will help you understand the countercurrent exchange. All right, so let's do a multiple choice question about this. And again, you can send me your answer to this question in a private chat to me. Countercurrent flow is more efficient in exchange than a concurrent flow. A concurrent flow would be if you have two fluids going in the same direction. So countercurrent flow is more efficient than concurrent flow because countercurrent flow provides what? Okay, 5 more seconds to get your answer in, A, B, C, D, or E. And I got lots of answers. I got all 5 of these answers from you guys. The most common answer that I got was C, but I don't know that half of you got that. But that was probably the most common answer that you gave me. And so I want to go back to this picture and just show you that all along this membrane there is a gradient, and it's not a strong gradient, it's a weak gradient. So it's 150 to 140. So that's not a big difference, but all along this exchange surface there is a gradient, so that there's diffusion all along there. So the correct answer is C, that all along the capillary there is a weak concentration gradient, a difference in, and I probably should have said partial pressure gradient rather than concentration gradient, but a weak gradient of higher oxygen in the blood and lower oxygen, sorry, I said that backwards, higher oxygen in the water, lower oxygen in the blood. So the oxygen's going to diffuse into the blood all the way along that capillary. Okay, now, if gills are so great, and they're huge surface area, so why can land animals not use gills, except for maybe those octopi that are able to go on land and survive for a little while? So why can't most land animals use gills? Send your answer to your lab partner in a private chat. What keeps land animals from using gills? I guess another question could be, why do you have to be in water in order to use the gills? Okay, so I'm going to call on Caroline.
So we said that air like doesn't flow the same way that water does, so it'd be kind of useless, and that gills are specialized to extract oxygen from water, not air.
It's certainly true that gills are specialized to extract oxygen from water. I would say that air is completely able to flow. Air flows even more easily than water. So it's not an airflow issue, although that was a good guess. So let me call on Danny. Oh, let me call on I think Danny's not with us. How about Mayela? Oh, Danny, you are with us. Go ahead.
Are you able to hear me now?
Yes.
Okay, I just said because part of their job is like extracting water, and we have like lungs for a function that's just focused on air.
So I think maybe your answer reflects a very common misconception. So gills are they're not extracting oxygen from water molecules. There are oxygen molecules that are mixed in with the water molecules. So it's not like they're tearing the water molecules apart. And maybe that's not what you thought, But really the gills are just for diffusion, which is the same thing that the lungs do. So let me call on, how about Kaylin? What would you say?
I said that the gills must be surrounded by fluid to be moist, and being on land would dry it out. And then also, like, the gills have more surface area, so each gill is surrounded by the fluid.
Okay, so you can have a lot of surface area in lungs too. So it's not so much the surface area, but your first answer is absolutely right. And that is that the gills would dry out if they were not surrounded by water. And also the gills would collapse because they rely on the water to kind of support them. And so you can just sort of imagine if you take the gills out of water, how they would all just collapse. And so then they would lose all their surface area if they did so. So you could an animal could probably keep its gills wet, but then the animal would lose so much water from its body that that just wouldn't be able to survive. So because remember that the gills are out foldings of the body, and so they're just exposed to the water. If they were just exposed to the air, they would dry out pretty well. Okay, so that's the main thing, is that they need to be wet. They need to be surrounded by water in order to sort of maintain their structure. So animals that are on land, instead of having gills, they have other ways of doing gas exchange. So in insects, they have tiny little air tubes that branch all over the body. And these air tubes are called tracheae. And so you can see these, there are external air openings. They're actually called sphericals, all along the abdomen of this grasshopper. And so air goes into these tubes and it fills these air sacs. And then all the body cells are either close to one of these tiny little air tubes or they're close to these air sacs. So they actually don't rely on their blood for gas exchange. Remember that they have an open circulatory system. So the blood would, or their hemolymph is what we should call it, would be kind of bathing all of these cells. But really they rely on these tiny little tubes all over the body to supply the air. So this is, you can see that there's a lot of surface area here. So it's not, the reason for using tracheae instead of using gills is not a surface area thing, because they've got a lot of surface area with all this branching of these tracheae, but it's because since it's internal, it's not going to dry out. Most land animals have lungs, which again is an infolding of a body surface that's divided into many tiny pouches. And again, that's where you get the surface area, is dividing it into many, many tiny little pouches. So in your assignment for tomorrow, I will ask you to label the bronchioles, larynx, pharynx, trachea. Since we only have 4 minutes left today, I will not throw you into a breakout room and have you do that. But basically, we already learned about the pharynx. The pharynx is the throat, and it's what connects the air system with the food system. So both the food passes through the pharynx and also the air goes through your nasal cavity and through the pharynx. This is also why you can breathe through your mouth, because the pharynx connects both. And then the food is typically going to go through the esophagus, which is behind the larynx. So you can feel your larynx in the front of your neck. You can feel the cartilage on the surface of your larynx. The larynx is also known as the voice box. And if you're talking, and if you put your fingers on the front of your larynx, you can feel vibrations. So your vocal cords are inside your larynx. And then air goes from the larynx into the trachea. The trachea divides into a right bronchus and a left bronchus, and each of those bronchi divides into smaller bronchi and then eventually into smaller bronchioles all over the lungs. And each bronchiole eventually divides into a bunch of sacs that are called alveoli. And notice that the alveoli are surrounded by blood vessels. And so this is where the exchange of gases happens between the air in these alveoli and the capillaries that surround them. Okay, so we're going to stop there, and we will pick up on the respiratory system on Friday. So you guys have a great day. And if you have any questions, hang around.